This video is about translation. Remember, translation is part of the overall process of gene expression, where we take a gene and use that information to make a protein. This happens in two basic steps, where we take the gene made out of DNA, we do transcription to result in RNA. Now that we have that RNA, we can use it. There's two types, messenger RNA and transfer RNA, that we'll use at a ribosome to make a protein, and it's this process that we'll focus on today, the process of translation. So translation is more complicated than transcription. Translation, remember, the DNA is kept safe and sound in the nucleus. The RNA exits and goes to one of these ribosomes, either free-floating or on the rough ER, and that's where we're going to make the protein. So let's bring in some of our cast of characters here. First we have to bring in a ribosome, and that ribosome comes in a couple of pieces that stay apart until everything is ready. So in the initiation phase, I'm going to bring in my messenger RNA. So I might note that this is messenger RNA. And we're going to start over here, a little different this time, at the 5' end, and move over. And once I got my messenger RNA, I'm going to put my ribosome around it and use the information here to make proteins. So the protein that I'll show you, I'm going to use this as a model. Remember, proteins are polypeptides made out of amino acids. And we want to string all these little monomers, these amino acids, together to make a protein. Now, that's the end goal, but how do we get there? How do we know which amino acids to choose? Because that matters a lot. Well, this is where our transfer RNAs come in. So here's my transfer RNA. You might know him as Percy, but he has a couple of parts to him that are important. One is the anti-codon, and the other is the amino acid that he carries. So here's our little bead that we'd used before to make that big string, but he's just carrying one of them, and this matching is very important. So here's our tRNA. Now, these are both parking spots in a sense, and we always start here. So kind of like in a company, if you have parking spots and you have your name on them, well, here's the name. This right here is a codon. And a codon is a set of 3 nucleotides on the messenger RNA. It's important you remember it's on the messenger RNA, that's the codon. Because this, he knows that he can park there because he has the complementary set of nucleotides to that. Complementary to A is U, complementary to U is A, and complementary to G is C. So those are complementary. He can park here because he has the right anti-codon that's complementary. All right, so we got one person position. This, by the way, is called the anti-codon. So codon on the messenger RNA, anti-codon on the tRNA. Nothing much happens yet until the next tRNA comes into place with the corresponding anti-codon to this codon. Again, each of these are complementary to each other, so he can park there. Now that they're together, this is what the ribosome does, and I won't go into the details of this, but this amino acid can be linked. Oh, these guys are falling apart on me. To this one. And now this guy can go off and find another amino acid, in this case methionine, that he can use and bring back to the ribosome in case more methionine is needed. Now this is kind of the weird part that doesn't quite work in reality, which is this is going to shift, so he needs to park over here now. Now we have a free parking spot, so we need another amino acid, and we need an anti-codon that matches. So complementary to AAA is UUU. They can park there. And this growing chain of amino acids can now be transferred to this tRNA, and that slot in the ribosome, that's empty. It's going to drive off. This is going to shift over, which means that he needs to move over. And we just keep going. So this is the elongation phase. Bring in yet another one just complementary to the next string. This gets transferred over. That pulls out, and so on and so forth. Let's get one more in there. So there he is, and finally transfer on this one. Now this is going to keep going and going in the same process, this elongation phase. But eventually, and this would go on for quite a while, we'd usually have dozens, if not a couple hundred amino acids to make a protein. But eventually, we will reach the end of the line. And when we reach this special codon, there's 3 of them, called a stop codon, then we get a special tRNA that comes into place. And his job is to say, stop, you're done making, there's a little more RNA past here, so we need to know where to stop. And this one is going to say, stop, we're done. And that means that messenger RNA can leave, the ribosome can fall apart, this guy can leave, and most importantly, our little polypeptide that we just made, isn't he cute, is all done. He can then be folded up, and to put it back in the context of the cell, this just got, not quite to scale, cranked out of a ribosome somewhere, and it probably float through a vesicle to the Golgi apparatus, and then that will be routed out maybe to be a receptor in the membrane, or maybe out of the cell altogether. That's the process of translation, making proteins that eventually end up creating what we might call our phenotype.